Well, good afternoon. Welcome to the podium session 11 on inner ear therapeutics. My name is Wei Chen, and I'll be moderating this session along with my co-moderator, Dr. Gwen Jeliak. As a reminder, each speaker will have 15 minutes for each talk, and this will include the questions. We would like to keep the sessions on time if possible. There are a couple of announcements I would like to make. First of all, Dr. Cynthia Morton will be giving the first talk instead of Dr. Hila Romi. And the second talk by Dr. Chong Kui will be a pre-recorded talk due to the time difference. And also the fourth and the fifth talks of the session given by Dr. Danielle Lenz and Dr. Michelle Valero, respectively, will not be recorded. So with that, let's get the session started. The first talk of the session will be given by Dr. Cynthia Morton, and she'll be telling us about a new mouse model of DFNA9. So thank you.
First of all, I'd like to thank all the organizers and individuals who made this meeting possible, because I can't imagine the number of sleepless nights that must have gone into it. So thank you very much. And I'm really privileged to make this presentation on behalf of Hila Romi, who's a clinical geneticist and postdoctoral research fellow in my laboratory. Hila's mom passed away at the end of the week, last week, and she notified me that she would not be able to make this presentation. And in her email, she told me that my mom worked so hard all her life to allow me to get educated and provide me these opportunities, which she didn't have. And so I think I like to give this talk in loving memory of her mother, Esther Romi. So our goal is to develop a molecular therapeutic approach to treat an autosomal dominant hearing loss caused by pathogenic variants in the COAC gene, which is associated with DFNA9. Just a little bit about COAC. It's the acronym stands for coagulation factor C homology, and that reflects the fact that a domain in this protein is actually evolutionarily conserved back to Limulus, which is the horseshoe crab. So the gene is highly expressed in the cochlea and encodes the protein coquelin. Coquelin is a large secreted protein. It's abundant in the extracellular matrix of the inner ear. It's expressed in the fibrocytes of the spiral limbus, the spiral ligament, and the stroma underlying the cochlear and vestibular sensory epithelial tissue in mouse and human. And the function of the gene is thought to be in an immune response. This is just a schematic representation of the protein that is designated with known pathogenic variants. There are presented here 29 variants, 27 of them are missense variants, and 2 are in-frame deletions. The variants are clustered in the LCCL domain, and that is the Limulus homology domain, as well as the second von Willebrand factor A domain in the 3' region of the protein. The protein has secreted has a signal peptide, and then the other domains that I just mentioned. So dominant gain of function pathogenic variants in COAC are causative of the adult onset progressive sensorineural hearing loss with or without vestibular dysfunction, known as DFNA9. The dominant negative effects of COAC pathogenic variants are attributed to the gain of a deleterious function of coquelin, where coquelin either aggregates and then causes deposit formation. Individuals harboring pathogenic variants in the LCCL domain in the 5' region of the gene are characterized by late onset in the 5th to 6th decades of hearing loss with vestibular dysfunction. And those who have variants in the 3' region of the COAC gene have an early onset 2nd to 3rd decade hearing loss without vestibular dysfunction. So the dominant disease mechanism as a result of the COAC mutations is particularly amenable to the proposed plan for gene targeting. By eliminating the pathogenic variant through a gene therapy strategy, we can prevent the aggregate formation. That's our goal. And haploinsufficiency does not result in disease. The preservation of the unaffected allele is sufficient for normal protein function. The disorder has a late, adult onset phenotype, so there's a window of opportunity for intervention. And this is a non-syndromic condition affecting only the inner ear, so it lends to local versus systemic therapy of CRISPR reagents. Our main goal, again, is to develop a molecular therapeutic approach to treat an autosomal dominant hearing loss caused by COAC pathogenic variants associated with DFNA9. We are utilizing a CRISPR-based gene editing strategy to disrupt selectively the pathogenic COAC allele with the other intact functional wild-type allele remaining that would allow proper hearing function. The studied missense variant causing the change that's shown here in the protein will be an alanine change to threonine at position 449, is in the 3' region, as you can see, in the second von Willebrand factor A-like domain. The phenotype of the individual with this change is moderate to severe progressive hearing loss, onset in the 2nd to 3rd decade of life, and no vestibular dysfunction, which is consistent with the individuals with pathogenic variants in the 3' region of the gene. Examination of this region of COAC revealed a PAM site for a number of Cas9 nucleases, and of these, the Cas9 KKH is an ideal choice for disrupting the variant because the PAM site is created at the position of the variant, and the enzyme should therefore cleave and disrupt only the variant allele with the normal allele remaining intact. So one of the first things we did was to do some in vitro experiments by establishing a human dermal fibroblast cell line from an individual harboring this pathogenic variant, and then co-transfect this with guide RNAs and the Cas9 KKH nuclease for targeting the COAC pathogenic variant. We now are basically transitioning this into the sort of in vitro experiments by creating skipped one slide. So the CRISPR gene targeting experiment, including the co-transfection, next-gen sequencing, and CRISPR analysis, did indeed show cleavage and indel formation only in the variant allele. So you can see the reference allele here at the top and the reads on the reference allele, and these are the reads that actually had successful allele-specific targeting of the variant. So the efficiency of targeting and disruption in the experiment was around 40%. So we're now moving to the in vivo experiments, and for this we created a humanized COAC knock-in mouse model, where in addition to targeting this pathogenic variant, we wanted to change the nucleotides in the mouse genome in the proximity of the variant to the human sequence that was found in the individual who had this particular variant. So you can see the sites that were changed actually to produce this particular humanized knock-in mouse model. And the mouse was created, born during lockdown, in COVID lockdown, and exhibited profound hearing loss. So you can see that the hearing loss was assessed by ABRs at various time frames from 3 to 4 months, then 5 to 6, and then later months. And there's a clear difference shown here between the wild type in this particular first graph from the heterozygotes and homozygotes at the 3 to 4 and 5 to 6 months, and at later months on the figure to the right. So of course we were very happy that the phenotype could be moved earlier, because paying the 4 seasons rates of mouse housing in Boston is not the best way to spend your money. So the Koch mutant mice exhibit profound hearing loss. In addition, we looked at DPOAEs, and they also were consistent with a profound hearing loss. And then we did vestibular testing as well, and you can see by these groups of sort of 3, again, the wild type het and homozygote, the phenotypes were actually consistent with a vestibular dysfunction. So this is fitting what the phenotype of the individual with the DFNA9. So in summary, we have the Koch pathogenic variant is targeted with this CRISPR-Cas9 genome editing methodologies in a skin fibroblast cell line from an individual with DFNA9. And the novel DFNA knock-in humanized mouse model was created. Then we are employing in vitro methods to evaluate targeting, efficiency, and specificity as a proof of concept for future in vivo studies of gene targeting and phenotype rescue in the humanized mouse model. So I'd like to offer a very special thank you to the individual with the pathogenic variant in Cochran that we've studied, who in this case literally had skin in the game. And on behalf of Gila and myself, I'd like to thank our colleagues both in Boston and in Omaha who contributed to this work, and also the NIDCD for supporting the studies of DFNA9, including an F32 that was awarded to Dr. Romy, and to private philanthropy to make this research possible. So thank you very much.
Thanks so much, Dr. Morton. So we have a couple of questions in the Q&A. So the first question is about the evidence supporting the absence of vestibular function in some of the DFNA9 patients. Were they examined specifically, or was it just from patient history that they didn't have any vestibular dysfunction?
There have been, in some instances, there have been rigorous testing for the phenotype, and it is very consistent among those that are tested. So you see this real split of the 5' variants versus the 3' variant. So I don't really have an explanation for why that's the case. So it's like either you have to take getting the disorder, the hearing loss later, and then also with vestibular disorder, or getting the hearing loss early and no vestibular disorder.
And there are a couple of questions about whether these patients have any bleeding problems.
I'm sorry, can you repeat it?
Oh, I think there are a couple of questions about whether these DFNA patients have any bleeding disorders, coagulation disorders.
Oh, no, not to my knowledge.
And then there was another question about the whether you guys tested the hearing thresholds of these humanized mouse without the A449T mutation.
So, I mean, there were wild type mice also, you know, in each litter. So these were, if I am understanding the question correctly, yeah.
Yeah, that's my guess in terms of what this person is asking. So, well, thank you so much for the great talk. And let's move on to the next talk. The next talk will be given by Dr. Chung Kui, and this is a pre-recorded talk, and she will be telling us about the application of CRISPR genome editing to a mouse model of DFNA2.
Hello, everyone. My name is Chung Cui, a doctoral candidate from Eye and ENT Hospital of Fudan University, Shanghai, China. It's a great honor to share my work with you today. The title of my project is High-Efficiency Genome Editing in Hair Cells Improves Hearing in a Mouse Model of Dominant Deafness. Next, I'll introduce the following 4 sections about this project. The first one is background. Hereditary deafness is a common genetic perceptual defect in humans. According to WHO, more than 1.5 billion people live with hearing loss, and 34 million children have deafness or hearing loss. 60% congenital deafness is associated with genetic factors, and DFNA2 caused by KCNQ4 is a common autosomal dominant deafness. Hereditary hearing loss is mostly a monogenic disease, and more than 120 pathogenic genes have been identified. And the cochlear tissue is small, and fewer cells needed to be targeted, so hearing loss may be easily rescued by gene therapy. In addition, there are no effective therapeutic drugs available, so it's appropriate to use gene therapy to treat hereditary deafness. CRISPR-Cas9 is a novel gene editing technology that allows efficient and specific knockout of genes, and now is being applied for the treatment of deafness, and has successfully improved hearing loss caused by TMC1. Our team also used CRISPR-Cas9 to rescue hereditary deafness caused by MEL6 and acquired deafness related to HTRA2. KCNQ4, a voltage-gated potassium channel, is mainly expressed in the auto hair cells. KCNQ4 mutation leads to dysfunction of potassium channel protein and result in DFNA2. According to the previous study, the normal hearing in KCNQ4 knockout mice suggests that we can specifically disrupt the KCNQ4 mutation allele to prevent or treat DFNA2. CRISPR-Cas9 has shown great potential in application for hearing loss gene therapy, but the low editing efficiency of in-ear in vivo makes it difficult to accurately assess therapeutic efficacy. So, based on the above research background, we propose 2 scientific hypotheses. Number 1, could gene therapy of knockout be used to precisely rescue hearing loss caused by KCNQ4? Number 2, is there a method to accurately detect the editing efficiency in hair cells? Firstly, we identified a novel KCNQ4 pathogenic mutation in a large Chinese step family. The hearing threshold of the patients show a delayed and progressive deafness. Then we established a mouse model with a humanized homologous mutation of KCNQ4. We measured ABR and DPOE, found that the mice also showed a progressive hearing loss. Red, blue, and black represents homozygous, heterozygous, and wild type, respectively. These results consistent with the hearing characteristics of the patients, indicating that we successfully constructed a mouse model that mimics the deafness caused by the KCNQ4 mutation in the patients. To explore potential treatment strategies for deafness by CRISPR-Cas9 technology, we designed 4 siRNAs of SA-Cas9-KKH against the Cas9q4 mutant allele. To screen an efficient and specific siRNA, we tested the editing efficiency of siRNAs by transfecting plasmids into skin fibroblasts derived from Cas9q4 wild-type, heterozygous, and homozygous neonatal mice. And the editing efficiency of GADR3 was the highest in Cas9q4 homozygous and heterozygous fibroblasts, while no obvious editing efficiency was observed in wild-type fibroblasts. For siRNA3, deletion mutations caused frame-shift mainly. All these results in vitro suggest that SA-Cas9-KKH GADR3 performed best, and GADR3 was used in subsequent experiments in vivo. This is the experiment protocol in vivo. SA-Cas9-KKH GADR3 was packaged into AAV-PHP.8b, which could infect hair cells with high efficiency in our previous work. We injected the treatment system into the inner ear of the neonatal mice in postnatal 1 to 2 days and assessed the hearing function at 8 to 12 weeks. Our study shows a positive correlation in editing efficiency and mouse age. However, the actual editing efficiency of hair cells remained unknown. To accurately assess the editing efficiency of SA-Cas9-KKH GADR3, Cas9q4 homozygous mice were mated with 801 GFP mice to obtain offspring, in which hair cells was specifically labeled with GFP. We injected the treatment system into the inner ear of offspring obtained by cross. By FCS and NGS, we observed average 34% indel frequency from the GFP-positive hair cells after sorting, and with a high efficiency up to 54%. And no obvious indel was observed in uninjected groups. These results indicate that a method was successfully established to accurately test the editing efficiency of the CRISPR-Cas9 system in hair cells, and demonstrated that the target DNA in hair cells could be edited with high efficiency by CRISPR-Cas9. We measured ABR and DPOAE to assess the therapeutic efficacy on auditory function in Cas9q4 heterozygous mice after treatment at 8 and 12 weeks. Red represents uninjected ears and blue represents injected ears. We observed lower ABR and DPOAE threshold in the injected ears than the uninjected ears, revealing a significant improvement of hearing function of mice. We also observed higher amplitudes and shorter latencies of ABR-V1 in injected ears. In addition, we observed the number of the auto hair cells in injected ears were more than uninjected ears. And the auto hair cells of treated mice exhibit more hyperpolarized resting membrane potential than untreated mice. And then we employed the circle-seq method, an unbiased and highly sensitive off-target assay in vitro to identify potential off-target editing sites, and the results showed only one candidate off-target site. The off-target site 1 identified by circle-seq, and the off-target site 2, 3, and 4 predicted by Cas-off-finder tool. Then, we performed these potential off-targeting sites in fibroblasts and hair cells by deep sequencing, and no obvious editing was observed. To evaluate the safety of the treatment system, AAV-SA-Cas9-KKH GADR3 was injected into the inner ears of the wild-type mice And we observed that the hearing of the mice was completely unaffected compared with the untreated mice. To exclude the possible effect of AAV or Cas9 protein on the therapeutic efficacy of casein for mice, we injected the AAV non-target system that did not target mouse genome into the inner ear of casein heterozygous mice, and observed that the hearing of mice was not improved compared to the untreated group. These results suggest that KCNQ4 mutant allele was specifically disrupted by the GADA3, resulting in the improvement of hearing in KCNQ4 heterozygous mice. Finally, we draw the conclusions. Firstly, we identified a novel KCNQ4 pathogenic variant from a large Chinese step family with autosomal dominant hearing loss. Secondly, we generate a humanized homologous mutation mouse model. Then, we improved the auditory function in KCNQ4 mice using the AAV-Cas9 KKH system. And then this is the first study to precisely test the editing efficiency of CRISPR-Cas9 technology in health cells. Firstly, thank my tutor, Professor Eli Shu, for his guidance and support. And thank Professor Huawei Li and Professor Ligenlin Li for the help for my project. And thank my team worker, Daqi Wang, Boy Huang, and Fang Wang. We worked together to finish this project. And I also want to thank the support of the following funds. Thank you. Wish you a happy Chinese New Year of the Tiger.
Thank you, Dr. Chung. So as I mentioned in the beginning of the talk, Dr. Chung is not here to take questions. So this is a pre-recorded talk. So, but if you like, you can still leave any questions you have for her in the Q&A chat box, and we'll be forwarding the questions to her. The next talk in this session will be given by Dr. Ksenia Aaron, and she'll be telling us about the selection of viral capsids and promoters on a TMPRSS3 gene therapy.
My name is Ksenia Aaron, and I'm currently in neurology faculty at the Cleveland Clinic. This work was culminated over the past 3 years while I was a T32 clinician scientist neurology fellow at Stanford. I would like to thank the ARO and all the panelists for the opportunity to present. I have nothing to disclose except that my collaborators are the inventors of AAV-KP1 capsid that I'll be talking about. Hearing loss in children, as mentioned, is a significant disease burden. Genetic mutations are a common cause of it, with more than 70% of all hereditary hearing loss attributed to autosomal recessive non-syndromic deafness. When looking at hereditary causes of hearing loss, transmembrane protein serine 3, TMPRSS3, makes up to 10% of cases of non-syndromic deafness in some populations. This mutation in humans has both DFMB10 prelingual and 8 postlingual variant. The protein contains 4 domains and has 453 amino acids with structure highly conserved between humans and mice, having 88% identical amino acid sequence. A group out of France was the first one to study a mouse with this genetic mutation. They showed that TMPRSS3 mRNA was detected in sensory and supporting cells, lesser and greater epithelial-rich, stria vascularis, and spiral ganglion neuron. They used a mutant mouse carrying nonsense mutation in exon 7 and showed that these mice were deaf as measured by ABR at P60. Scanning electron microscopy seen here demonstrated degeneration of the organ of Corti of both inner and outer hair cell, which began at around P12 in the basal churn and progressed toward the apex, reaching completion within 2 days. Nevertheless, not a lot is known regarding the function and the mechanism of this gene within the inner ear. So why study this genetic mutation? For one, it seems to affect various cells in the organ of Corti, and two, there is a potential of hearing restoration on a multicellular level. Given the preliminary studies, our hypothesis was that gene therapy can prevent cell degeneration and hearing loss in the TMPRSS3 mutant mouse model. So our first goal was to study and understand the kinetics of this mutation by characterizing a novel mutant TMPRSS3 targeted mutation 1 Lexington mouse, knockout of which was generated by introducing a stop codon between exon 1 and exon 2. We examined several time points, first confirming expression of this gene, then evaluating what happens at the onset of hearing, as well both in acute and long-term time frame. In situ hybridization at P5 demonstrated that indeed the mRNA of TMPRSS3 is seen in both inner and outer hair cells, supporting cells, lesser and greater epithelial-rich, interdental cells, and spiral ganglion neuron. We confirmed our mutant mouse model on the lower panel, as you can see, by absence of mRNA signal in the organ of Corti as compared to the wild type seen in the bottom panel at P1. At the onset of hearing, the mutant mice had lost more than 99% of both inner and outer hair cells that occurred between P14, which is the middle panel here, and P14 and a half, bottom panel, much shorter time frame than previously reported. This was seen across all 3 turns of the cochlea simultaneously. ABR measurement at P21 demonstrated that indeed our mutant mice were deaf as compared to its legitimate control. We then quantitatively analyzed the changes that happen over time to cells that express TMPRSS3 mRNA. Looking at counts in whole mount tissue prior to cell death at P12, we found that counts of both sensory myo7 positive cells on top panel and supporting cells SOX2 here on the bottom panel in mutants were similar to its wild type and heterozygote littermates. At P21, as expected, both inner and outer hair cells were absent across all 3 turns in the mutant mice, while supporting cell counts were the same, albeit more disorganized. Looking long-term, we saw a decrease in supporting cell counts in mutants by P120. We additionally observed a significant decrease in spiral ganglion neurons by P120 as well. So now that we have a mouse model, we wanted to see if we can rescue this cell degeneration and restore hearing. We know that several recent studies have demonstrated success of gene therapy in the inner ear, with high transduction efficacy of various capsids on multicellular level that has been achieved. However, it remains unclear whether high transduction is truly necessary within the inner ear, especially when you target an inner ear disease model that affects multicellular level. So our second goal was to study whether cell degeneration and auditory changes in our mutant mice can be prevented using gene therapy. We did this first by testing a novel AAV capsid to assess transduction efficacy in the inner ear, and second, creating a TMPRSS3 AAV vector and testing both in vitro and in vivo. In collaboration with my co-mentor, Professor Mike Kay, his laboratory recently developed a novel capsid, AAV-KP1, which was shown to have high transduction efficacy across multiple cell lines, both mice and humans, compared with other capsid. Using a TD tomato expressing KP1 capsid under cat promoter, we injected 1 microliter at P1 and then in through the posterior semicircular canal, and then performed analysis at P21. What we saw is that P21, a high transduction efficacy of the organ of Corti was achieved for both sensory and supporting cells, and the capsid itself did not affect the EBR as compared to the control saline injection. We then obtained a TMPRSS3 clone from origin and created a cat promoter KP1 TMPRSS3 vector. When examining transduction and transfection efficacy in vitro, in both LN293 cell lines, we noticed that the cell in the flask looked kind of sick shortly after plating and had to be harvested much earlier. The cells appeared to be detaching, especially at the higher multiplicity of infection. MTS assay was then done to look at cell viability, which demonstrated that with higher MOI and prolonged incubation, the cells were dying. We also simultaneously injected the KP1 vector with TMPRSS3 gene into our mutant and wild-type mice. In wild-type mice, what we observed was that there was some cytotoxicity on the animals, as can be seen here at P21, with disorganization of the inner and outer hair cells across all 3 turns of cochlea. The ABR also showed significant threshold shift compared to saline-injected animals, with significant quantitative decrease in myo7A cells as compared with control. There was no effect on supporting cell counts, which is not demonstrated here. We then decided to evaluate, well, what if we use dilution of the titer and would it reduce the cytotoxicity effect in the wild types? So at full titer shown before, we saw that there is a loss and disorganization of the sensory cells, which you can see here in the top panel. We then did a half dilution, and quantitatively it did abolish the sensory cell cytotoxicity, but inner hair cells still appeared more swollen rather than flask-shaped. With 1 to 10 dilution, the cytotoxic effect was completely abolished histologically in the wild-type mice. Neither of the titer was able to restore histology in the mutant mouse, as can be seen here in the bottom panel. Histological changes also correlated with ABR, with the full titer here demonstrating that there was a significant ABR threshold shift across frequencies in wild-type animals, black box, as compared to saline-injected green triangles. There was still ABR shift in the subgroup of the wild-type animals 1 to 2 dilution, likely correlating to more swollen inner hair cell state. And the ABR thresholds were comparable to saline-injected animals at 1 to 10 dilution. However, neither of the titer was able to restore ABR in mutant animals, represented here by red circles. Well, we then thought, well, maybe our capsid had high transduction efficacy is the reason for cytotoxicity of our exogenous TMPRSS3. So we decided to evaluate whether using a different, less efficacious capsid was going to have a decrease in cytotoxicity effect on the wild-type animals, and at the same time restore degeneration effect in mutants. We used AAV-DJ under CAC promoter and saw that it had less transduction of various sensory and supporting cells of the organ of Corti without affecting the ABR. We then used TMPRSS3 carrying DJ vector and saw that while histology was not affected, there was still evidence of significant ABR shifts across several frequencies in the wild type, with no evidence of rescue in the mutant mice. So at this stage, we saw that dilution and having a less efficacious capsid can decrease cytotoxicity of our exogenous TMPRSS3 in wild types, but still unable to rescue our mutant mouse. Based on some literature from the ophthalmology world of gene therapy, we decided to evaluate whether using a weaker promoter instead of CAD will resolve this. So first, we evaluated transduction efficacy of using a weaker promoter elongating factor 1, EF1 alpha, in KP1 vector. At P21, while both outer and supporting cells maintained their high transduction efficacy, there was almost no transduction detected in the inner hair cells with TD-tagged capsid alone, and as previously shown, it did not affect ABR thresholds. TMPRSS3 carrying KP1 vector under EF1 alpha promoter did not demonstrate any cytotoxicity in wild-type animals at P21, both histologically or in ABR, as will be seen later. As shown here, in the injected ear of mutant mice, we saw prevention of cell degeneration that was observed both short-term at P21 across all 3 turns, and long-term at P120 with counts comparable of that to the wild-type littermates. We also observed the significant restoration of the spiral ganglion neurons at P120. ABR thresholds, while not fully restored in the injected animals, seen here as red triangles, were able to get partial threshold rescue across several frequencies, albeit you can see here it was with lower amplitude at both P21 and it was maintained by P120. So in summary, AAV-KP1 capsid had high transduction efficacy in murine cochlea. Our gene transfer of TMPRSS3 under a strong CAC promoter led to cytotoxic effect both seen in vitro and in vivo. We were able to ameliorate the cytotoxicity of the exogenous gene in wild-type animals by either reducing the titers or the transduction efficacy, like using AAV-DJ. But none of these methods were able to restore the mutant degeneration. And it was only through using a weaker promoter, EF1 alpha, that we were able to do both, ameliorate cytotoxicity in wild-type animals and restore mutant degeneration. Finally, I would like to thank all of my mentors and lab members that contributed to this project, specifically Dr. Ching, who was instrumental in my success over the past 3 years, as well as all of my grant support and my collaborators. And again, thank you to the AORO for this opportunity to present, and I will take any questions if there's time.
Thank you, Dr. Aaron, for that beautiful talk. We have a couple of questions in the Q&A chat about the viral titer that was actually delivered in these animals.
What specifically is the question? I'm sorry.
Oh, do you know the actual viral titer that was used for both the AAV CAG TMPRSS3 and also the ones with the EF1 alpha promoter?
Yes, it was 2.0 times 10 to the 12 viral VGs per milliliter. And we try to maintain the similar concentration between stronger and weaker promoter, as well as using just TD tagged capsids, so it would be representative across all.
And the dilution that you guys used was the same volume, is that correct? Essentially,
yes, it was 1 microliter, so then it would be 1 times 10 to the 12, and then it would be 1 times 10, 2 times 10 to the 11. I mean, just the serial dilution, yeah.
You know, it's fascinating, you know, with the, you got such wonderful results with the EF1 alpha promoter where it causes a lower level expression. You would think that you would see similar results with the, you know, 1 to 10 dilution potentially, but I wonder if there are cell types that express PRSF3 that's seen in the EF1 alpha promoter that you're not seeing. I'm sorry, that's expressed by the CAG that you're not seeing in EF1 alpha promoter, or do they express the
we
same
not shown in this presentation, but we looked at TD tagged at earlier time point, both P6 and then at P21. We actually saw a difference in CAG versus AF1 alpha. With CAG at P6, it was not highly transduced in inner and outer hair cells, but very highly transduced in supporting cells. And by P21, it picked up across all cell types, at least in the ones that we looked at, the inner and outers and support and various supporting cell types. Whereas with EF1 alpha at P6, we actually saw a strong through both supporting and sensory hair cells. So both inner and outer hair cells had almost 100% transduction efficacy, as well as supporting cells. And as shown here, by P21, our inner hair cells almost had no transduction. So there is some difference in how CAG and EF1 alpha are, I guess, turned on in a way, or like promote, you know, a switch of the gene to be expressed within a different time point, kind of. Yeah.
There is a question about whether you tried other AAV capsid like PHPV or NKD.
No, just because my collaborator invented both DJ and KP1, so we knew that DJ, and because they compared DJ and KP1 already and had a very good data out of that, so we knew that DJ was overall across various cell lines much weaker in efficacy, so we decided to use DJ.
Mm-hmm. And there was another question about if you had looked at the infection efficiency of the spiral ganglions with the AAV KP1 versus the AAV DJ.
So I did specifically look at KP1, and we had a basoapical gradient where apex was highly transduced in spiral ganglion, but it was still not 100%, it was about 60%. And then by base, it dropped off to 30%, and that was with CAG. We did not specifically look with EF1 alpha and our, yeah.
Mm-hmm. And then there's another question about whether you looked at the EP in these animals.
We did not. They did, the French group did look at EPs. We did not look at our EPs of these animals.
Mm-hmm. Okay. Well, it's a beautiful talk. Thank you so much.
Thank you.
I think we need to move on to the next talk.
So the next talk is from Marina Ivanchenko from Harvard Medical School. And the title of her talk is Dual AAV PCDH15 Gene Therapy for Usher Syndrome Type 1F Deafness and Blindness.
Good afternoon, everyone. Today I will be presenting you our study development of dual AAV PCDH15 gene therapy for Usher syndrome type 1F. You know that hearing happens when sound waves delivered to inner ear in the form of fluid vibration and change to neural signals inside the cochlea. And hair cells in the cochlea are responsible for amplifying detecting sound. And in this cell, PCDH15 protein binds to CDH23 protein to form tip link between stereocilia, pulling directly ion channel to initiate the electrical response. In photoreceptor cells, studies in human, in non-human primates, and amphibians, they shown that all Usher proteins are located in the calyceal processes. This is actin field protrusion that emerge from the inner segment of photoreceptor and form like barrel-like structure toward the outer segment of photoreceptor in cones and in rods. And Christine Petit's group, using electron microscopy, shows that these processes connected to outer segment membrane by link composed with PCDH15 protein. Such ring-like structures or calyceal process is absent in photoreceptor for rodents. We know this. We also know the structure of PCDH15. It is a large protein, more than 200 amino acids. It forms 11 EC domain repeats and transmembrane domain. In human, mutation of this protein causes isolated deafness or Usher syndrome type 1F, which manifests as profound deafness, as balance deficit, and blindness development over several decades. Currently, treatment for Usher 1F for deafness is limited for cochlear implants, and unfortunately, there is no treatment for blindness. So gene addition therapy could be attractive treatment for those patients with homologous recessive mutation. We know that mouse models for Usher 1F show profound hearing deficit, balance deficit, but little or no vision deficit. So in this study, we tested our approach in 3 different mouse models. In 3 different models, 2 of the mouse model, we restored hearing in conditional PCDH15 knockout mouse. We restored balance in constitutive PCDH15 knockout mouse. And we also established proof of concept in human retinal organoids to prevent blindness. The problem with PCDH15 is the coding sequence for PCDH15 is too large to fit into single AAV into one capsid. So one strategy that we explored is to engineer 2 vectors. So each vector as a part of full-length PCDH15 to produce full-length mRNA in cells by both vectors. So one vector includes CMV promoter, N-terminal of PCDH15, splice donor site, and highly recombinogenic sequence. And then the second vector, again, highly recombinogenic sequence, splice accessory site, and C-terminals of PCDH15 and other regulatory elements necessary for the expression. So once AAV vectors are in the same cell, the HR sequence recombines to create artificial intron, which splice out to create full-length PCDH15 coding sequence. And firstly, we tested this approach in HEK cells that have no native PCDH15 expression. So we infected them with AAV9-PHPV that carry both those vectors and infected those viruses with HEK cells. We collected mRNA, then mRNA harvest, reverse transcribed, and we sequence cDNA library around the splice junction site to confirm the proper recombination and splicing in HEK cells. First, our aim was to restore hearing. And for this, we generated PCDH15 flocked mouse line created by LOX-P site around exon 31, which is encode critical transmembrane domain. This mouse was crossed with PCDH15 CRE mouse, and which CRE recombinant gene expression driven by myosin 15 promoter and delete exon 31 at postnatal age after P0 after period of bundle development. And not surprisingly, scanning electron microscopy showed no Meyer abnormalities in these mice in P6 with preserved tip links. However, at P31, we've seen severely disrupted hair bundles with missing cilia, with missing hair bundles, or shortened cilia. When we did FM143 uptake to evaluate mechanotransduction function, we again didn't see any difference in the early stage, postnatal stage at P6, but we have not seen any FM loading at P35, suggesting that no function on tip links at a later age. ABR also confirmed that these mice are profoundly deaf at this age. So we packed design vectors into AAV9-PHPV. We injected mice at P1, and then we assessed ABR at P35, and then harvest cochlea for the histology. And we found that treated mice with dual AAV showed robust rescue of hearing assessed with ABR and with TPOE. And in the best cases, here is the red line, the ABR threshold was close to wild type level. We also perform immunofluorescence with actin labeling and scanning electron microscopy. And we observe robust rescue hair bundle morphology in treated animals compared to untreated animals. To detect exogenous PCH15, we design a tag in N terminal of PCH15, HA tag. So HA tag should not abrogate binding with CDH23 and should not change any properties of PCH15. So, but firstly, we designed the vector with HA tag and inject it into mice to test whether we can get the same level of rescue as a vector without HA tag. And as not surprisingly, the rescue of level was absolutely the same as with a vector that did not carry HA tag, confirming that HA tag is fully functional with PCH15. So then we perform immunofluorescence and immunogold labeling in cochlear-treated mice, and they display HA tag labeling located in the tips of stereocilia, and also gold beads in the tips of stereocilia were tip linked at that age. So can we rescue balance? For balance experiments, we could not use our conditional knockout mouse. It did not show severe vestibular phenotype. And we generated another mouse constituted a null PCH15 mouse. This mouse have no endogenous expression of PCH15 at any time through their life, including the critical stages of bundle development, which result in more disorganized hair bundles in the cochlea and in the vestibular system. These mice were also deaf with no detected threshold, but they showed severe vestibular phenotype, such as intensive head wobbling, circular behavior, hyperactivity, swimming difficulties, and they could not stay in a rotaral test you can see on this video. So now we injected constitutive PCH15 mice at P1, and we assessed vestibular function test at P30. And interesting that all tests that we perform, open field test, swim test, rotaral test, was fully restored to wild type level. And this image illustrates example of rescue of vestibular function in constitutive knockout mouse, which acts as normal wild type mouse. Now, what we can do with blindness for Usher1F? As I said, since mice do not develop choliceal processes in the photoreceptor, and mouse model of Usher1F show little or no vision deficit, our collaborators from Institute of Molecular and Clinical Ophthalmology in Basel developed human retina organoids from iPS cells and found that organoids mimic human retina. They develop a photoreceptor, they develop all three nuclear layers and synaptic layers, and even those photoreceptors can sense light. So then we ask question whether this photoreceptor in human retina organoids could develop choliceal processes. And as I said, we're interested in choliceal processes because as we looked on non-human primate and zebrafish, PCH15 is associated, PCH15 expression associated with choliceal processes, which kind of stabilize the outer segment of photoreceptors. So it was very important. So we looked into ultrastructure of photoreceptors in organoids, and it showed that majority of photoreceptors, they develop inner segment, they develop outer segment, and they and cilia, and we observe nascent choliceal processes and the apical end of photoreceptor, whereas normal PCH15 expressed. Then we asked what capsid we can use to deliver our dual AAV to organoids. We tried PH3B, EGFP, and we found that this capsid nicely transduce all photoreceptors in organoids. So we use this capsid to encode PCH15 CD1 isoform, deliver it, and immunofluorescence showed that HA tag localized in the surface of inner segment of photoreceptor and the inner outer junction site where cell processes develop. Then we did also immunogold scanning electron microscopy, and we found that gold beads with tag HA PCH15 associated with newly formed cell processes and also expressed and also located in the surface of inner segment. So altogether, this data suggests that dual AAV gene therapy for Usher syndrome is a promising approach for therapeutic gene delivery to human inner ear to rescue vestibular system, vestibular problem in patients, and also results are very encouraging for the treatment of the blindness. So I want to acknowledge people in my lab for their contribution. I want to acknowledge my mentor, David Corey, and our collaborators from Institute of Molecular and Clinical Ophthalmology in Basel, Usher 1F Collaborative, and Bertarelli Foundation. And my very last slide about announcement. We are very excited to tell that Molecular Biology Hearing and Deafness Conference that will take place this year in May in Iowa City. So please don't miss opportunity to send your abstract to present your work and attend this meeting and meet other scientists. Thank you for your attention, and I'm happy to answer your questions.
Thank you so much, Marina. Really beautiful work. There's a few questions for you in the Q&A. So Rona Erzano is asking, it seems that it takes a while for the tippling to lose functionality in the flux mice. Have you tried to inject the mice at a later time point, maybe P30 rather than P1?
For this experiment, we did not try to inject at P30 because this Myzin 15 conditional knockout mouse showed like no preserved hair cell bundle or almost missing all short and middle row stereotypical bundles at P30. So this mouse started to show a phenotype maybe at P15, severe phenotype. So that's why we started from P1. We did not try any later age.
Mm-hmm. Shahar Tabir is asking, commenting on nice results and asking, why did you use the conditional mouse rather than the knockout mouse?
We use conditional knockout mouse because it showed delay onset of Cree activity, because we wanted to start to test our approach and to start intervention when hair bundles preserved, when they are healthy. A next step can be like try to do intervention when hair bundles disrupted in the conditional knockout mouse, but chances will be lower because hair cells by the time is damaged. They are damaged.
Yeah, and that kind of answers the following question from Pranav Mathur, was asking if you've tested those dual AV in a full knockout mouse, but you pretty much answered it.
Yeah.
Let's see. Aziz says, great talk and data, Marina. Sorry, a vision question at ARO meeting. Have you tried with H1F organoids?
No, we have not tried yet. It's the next step. First of all, we need to evaluate whether H1F organoids have phenotype that can be rescued with our approach. But yeah, this is a very good question.
All right, one last question, and then I'll invite you to check the Q&A. From Jeff Ford, which protocatemin 15 splice form was encoded in your vector? Given the dual vector approach, might it be possible to encode all three non-splice form in a 3' vector?
In this study, we used for inner ear rescue CD1 isoform. Mm-hmm. For non-human primate organoids, we use CD2 isoform. I'm sorry, we used for inner ear, and CD1 we used for human retina organoids. It would be challenging to put all three because we know that CD1 isoform is very long, And I think we need to go with triple vector approach to encode all three isoforms.
Okay, great. Thank you so much, Marina. All right, we're going to move on to the next talk. Niliksha, sorry if I don't pronounce your name well, Niliksha Gunewardene. The title of her presentation is Supraparticle Mediated Neurotrophin 3 Delivery Preserves Drug Bioactivity and Promotes its Retention in the guinea pig cochlea. The podium is yours. Perfect.
Hi, my name is Nikki and I'm from the Bionics Institute in Australia. So as the last speaker of this session, I'll be switching gears and discussing with you some of the work that we've been doing on delivery of neurotrophins into the cochlea using a nanotechnology method. So to this audience, it is very well established that neurotrophins improve auditory neuron survival and also promote synaptic regeneration. However, variable outcomes and restricted long-term therapeutic benefits have impeded clinical translation. And it's possible that this is due to the short half-life of neurotrophins and also the rapid clearance of neurotrophins in the cochlea. As such, a safe, effective, and sustained neurotrophin delivery strategy is required. We at the Bionics Institute have been working on developing a system called supraparticles, which is essentially three-dimensional structures which is formed by the self-assembly of micro-nanoparticles. So this is an image of a supraparticle, and if you look onto the surface of these supraparticles, they're comprised of multiple micro-nanoparticles. The benefit of the system is that they permit long-term controlled release of drug, and furthermore, they're very advantageous because you can manipulate their size, structure, and function and tailor them based on their application, for instance, to deliver viruses or also drug. We have used these supraparticles and tailored them to deliver neurotrophins into the cochlea. Essentially, we have shown in a previous publication that over 45 days, you could release neurotrophins in vitro. In a subsequent study, we also showed that if you deliver supraparticles loaded with neurotrophins into a deafened guinea pig cochlea, you could improve auditory neuron survival across nearly all the turns of the cochlea. So based on these promising results, the next step of our study was actually to interrogate the bioactivity of the released neurotrophin and also analyze the pharmacokinetics of the released neurotrophin, both in vitro and in vivo. And so this brings me to the aims of this study, which was essentially to analyze bioactivity and also characterize the pharmacokinetics of the released neurotrophin. We tested two different approaches, the intracochlear delivery as well as round window delivery. And finally, we wanted to interrogate the safety of this platform. We used a published technique. Essentially, the bioactivity and the pharmacokinetics was measured using a radiolabeling technique. So the neurotrophin was tagged with a 125I isotope, which essentially enabled us to assess the bioactivity of the released neurotrophin, so we could track it. And also we could do a very thorough characterization of the amount of neurotrophin retained in the cochlea and also assess the biodistribution of the neurotrophin using both the intracochlear as well as the round window approach. This is an example image of an autoradiography of a whole cochlea of a guinea pig. And you can see that with delivery of just one supraparticle, there was widespread distribution of the drug throughout the cochlea. We initially needed to characterize these particles. So we needed to know exactly how much these particles could load in vitro. And so we did a particle loading test, and we loaded each particle with 7.5 micrograms of neurotrophin, and we found that on average, the particles could load around 5.5 micrograms of neurotrophin. I should also note here that each particle weighed around 50 micrograms. So this was essentially 10%. It was loading around 10% of the drug. In terms of drug elution, we looked at the elution profile of the supraparticle release neurotrophin, and we found that over a period of 30 days, around 70% of the drug was released, therefore indicating that there was a very sustained drug release over a period of time, over the period of time that we analyzed. We then went on to measure the bioactivity of the neurotrophin. So we used two tests here. We used a TRAC-C cell line as well as auditory nerve cultures. The TRAC-C cell line is a cell line that contains a beta-lactamase reporter gene, which is expressed upon stimulation of the TRAC-C receptor with neurotrophin. So expression of beta-lactamase is measured and calculated as a measure of neurotrophin activity. And so initially what we wanted to do was measure the EC100 or effective concentration of freshly prepared neurotrophin. And we found that with this dose response curve, we found that the EC100 was around 100 nanograms per mil. We subsequently used our supraparticle eluted NT3, and we cultured our cells with the eluted NT3 to assess the bioactivity of the neurotrophin that was released. And what we found was that over the time period that we analyzed, which was day 3 to day 28, so this was all in vitro eluted NT3, that there was well-maintained bioactivity of the drug, so indicating that the supraparticle eluted NT3 was not denaturing over time. We also validated this in auditory nerve cultures. So here, compared to no NT3 and then freshly prepared NT3, the supraparticle eluted NT3 had well-maintained bioactivity. There was a slight decline at day 28, but we think that this could be attributed to some variability also in our auditory nerve cultures. We then went on to do in vivo studies. So this work was done in guinea pigs. We have previously shown that when you deliver a bolus injection of neurotrophin, so here again we use this radio labeling technique. If you deliver a bolus of neurotrophin into the cochlea using the intracochlear approach, there's this exponential decrease in exponential drug clearance from the cochlea. And what we found was that after 3 days, there was only 1% of NT3 in the cochlea. When we looked at the autoradiography of the cochlear sections, you could see from 4 hours on to 14 days that there was this decline in drug distribution in the cochlea. We wanted to compare these results to our supraparticle eluted neurotrophin. So as I mentioned to you before, we used the intracochlear as well as the round window approach. In this slide, I'm showing you the intracochlear approach. So here a cochleostomy was performed and one supraparticle was implanted into the cochlea. Bilateral implantations were performed and we terminated animals at multiple time points. What we found, so this was on the y-axis is neurotrophin retention inside the cochlea. So these were gamma counts that were taken from the whole cochlea, which enabled us to calculate the amount retained. What we found was that after 3 days, there was around 55% of neurotrophin retained in the cochlea. So if you compare this to the previous slide where I showed you only 1% retention in the cochlea with a bolus of neurotrophin injection, this was quite a remarkable improvement in terms of the amount retained. We did a lot of autoradiography analysis, and here from 3 days on to 28 days, and you can see that there's this widespread distribution throughout the cochlea of neurotrophin when you deliver the drug through this intracochlear approach. Even at 28 days, there was some drug still present inside the cochlea. We then went on to look at it with the round window approach. So here, the round window was exposed and we used 5 supraparticles and they were placed within the niche of the cochlea. Again, bilateral implantations were performed. What we found here was after 7 days, so we had a slightly different timeline compared to the previous study, we had 43% remaining. Again, if you compare that to the 1% that was present in the cochlea after a bolus injection, here again, we were getting more neurotrophin retained inside the cochlea with the round window approach. I should note here that the gamma counts that we took of the whole cochlea was also the inside and also the outside of the cochlea, because the supraparticles were implanted just on the round window niche. With the round window approach, when we looked at the autoradiography analysis, it was significantly more variable. So drug delivery to the round window was the amount of drug that extended into the cochlea was significantly more variable with this round window approach. And a lot of the signal was coming mainly from the particle itself. However, even at 28 days, we could still see some drug present in the basilar membrane as well as Rosenthal's canal, where the auditory nerves are housed with this round window approach, which was quite promising. However, this was more variable than with the cochleostomy approach. We next wanted to investigate if there was any change in hearing thresholds, if the approach was safe. So with the intracochlear approach, here's an ABR graph of the intracochlear approach, and hearing thresholds were measured at 1, 2, and 4 weeks. You can see with the 1 week approach, there's a shift in hearing thresholds, and this could be attributed to the cochleostomy itself, some surgical damage. But at 2 and 4 weeks, there was a near return to normal, still a bit higher than the normal range, but a near return. With the round window approach, there was no real shift in hearing thresholds, indicating that the particles itself did not damage the hearing thresholds. We did also a thorough characterization of several organs to interrogate if there was any neurotrophin that was traveling to any of the other regions of the body, such as from the kidney all the way to the cochlear nucleus. With the intracochlear approach, the signal was primarily retained in the cochlea. With the round window approach, there was some in the bulla tissue, semicircular canal, and cochlea, but nothing present in any of the other organs, indicating a very good safety profile. So just to summarize, I hope I have convinced you that delivery of drug using supraparticles support continued long-term availability and release of biologically active neurotrophin in the inner ear. The neurotrophins are retained and effectively distributed throughout the cochlea using the intracochlear approach, but it was more variable using the round window approach. With both delivery approaches, there was a clear 125I NT3 signal in Rosenthal's canal, which contains auditory neurons, and these are the target populations of neurotrophins after neurotrophins. So this was very promising to us. And essentially, these findings indicate that supraparticles are a viable approach to effectively deliver neurotrophins to the cochlea for extended durations. Thank you. Finally, I'd like to acknowledge all those that have been involved in this project, particularly Professor Andrew Buys, Patrick Lamb, and Professor Rachel Richardson. Thank you.
Thank you so much, Niliksha. Beautiful talk. Let's see. We have some questions starting to come in. From Vikrant Bors. Nice work. Is there any immune response induced by these suprananoparticles? And what kind of supraparticles are you using for this study?
Yeah. So from our analysis of all the cochlear sections, we have not detected to date much of an immune response after the delivery of supraparticles. The kind of supraparticles we're using are made out of silica. Yeah.
Great. Let's see. What kind of hearing loss is selected for this treatment by neurotrophin? Age-related or noise-induced or drug-induced?
So multiple. It could be age-related or noise-induced. For the moment, like our initial indication would be to use it alongside a cochlear implant to reduce neural excitation thresholds. But yeah, we'll probably be looking into a lot of indications in the future.
Mm-hmm. A follow-up to this question by the same person, by Pudding, is metabolic presbycusis a good candidate for NGF? NGF or NT3, I guess that's what the question was. Metabolic presbycusis.
I'm not really sure about the answer to that question.
Yeah, I'm not sure either. Yeah. Let's see. Does the radio labeling affect the NT3 distribution in the inner ear?
Not to our knowledge. 125i is extremely small, which is why we used it. It does not influence the uptake across into the round window membrane either. It's a very small molecule that's attached to the NT3, so we don't think it impacts NT3 distribution in itself.
Okay. Another question from Dorothy Penn. What are the supraparticles made of? I don't think you had a chance to get into that.
Yeah, so they're made out of silica. Yeah. Okay,
so can we think of using this approach for other treatment, like antisense oligonucleotides, or any other? Do you think supra nanoparticle can be used for that?
Yes, definitely. So that's the main benefit of these supraparticles. They can be tailored, so their size, structure, and function can be tailored based on what you want. So yes, they can be used for delivery of siRNA, viruses, different drugs, and so they just need to be tailored according to the application.
Wonderful. Well, there are more questions, and we'll make sure to have the questions sent directly to you so you can answer them. I think I think everybody's ready to quit the Zoom format of this, and but it's been really a pleasure to host and co-chair with Wade this really interesting session. I thank all the speakers for great talks. Thank you for answering all the questions live and on a Q&A. And I look forward to seeing you again over the next few days, virtually and eventually in person.